Hi guys, welcome back to MedEd OTS, where we make medical laboratory science simple and easy to understand. So, in today's lesson, we are going to be talking about grain steaming technique, the importance of grain steaming, what is grain steaming, and why does the bacteria appear different in color after grain steaming. So, let's get started. Grain steaming technique is a technique that is used in the microbiology lab to be able to differentiate between microorganisms, right? So it helps to differentiate between gram negative. We have gram negative and gram positive organism. So gram staining technique is a technique that is used to be able to differentiate between bacteria, whether they are gram positive or they are gram negative. So it's very important. It's a very important technique in microbiology. So now let's understand the concept. It is based on the cell wall of the bacteria, right? So due to the bacterial cell wall, this gram stain will be able to penetrate. So we have two types of bacteria, as I said, right? We have gram positive and we have gram negative. So now let's talk about gram positive bacteria first. These are the properties of a gram positive bacteria. Gram positive bacteria. When we talk about gram positive bacteria, these bacteria have a thick cell wall. The bacterial cell wall is thick. They have a thick cell wall. Also, for gram-positive bacteria, they have a thick peptidoglycan layer. They have a thick peptidoglycan layer, right? And then another thing is that because they have a thick peptidoglycan layer, the gram stain, right, will be able to penetrate this peptidoglycan layer and also be able to stain the bacteria purple in color. So firstly, they have a thick peptidoglycan layer. So when they are stained with the gram stain, they retain a purple color. Remember this. This is very important. They have a purple color after staining. After they are being stained, they will retain a purple color because they have a thick peptidoglycan layer. A peptidoglycan layer, right? So this is one of the feature, right? One of the unique feature of a gram-positive bacteria. Another thing is that the gram-positive bacteria, they have no membrane. They have no outer membrane, right? There is no membrane here. So they retain the crystal violet stain. I will explain this in detail, don't worry. So they are purple in color after they are being stained. So let's do a recap before we go to gram negative. For gram-positive bacteria have a thick peptidoglycan layer, and then they do not have an outer membrane. And also, when you stain them with the gram stain, they retain the crystal violet color, which is purple. So under the microscope, they are seen as purple in color because they retain the stain. This is the unique So now let's talk about the gram-negative bacteria. For gram-negative bacteria, they have a thin peptidoglycan layer. Their own layer here is what they have thin peptidoglycan layer. Their layer is thin. And also another thing about them is that they have autocoring. Remember, I told you that for gram-positive bacteria do not have outer membrane, right? But for the gram-negative bacteria, they have an outer membrane. And then when you stain them with the gram stain, so for them, if you stain them with the gram stain, they have the pink color because they retain the counter stain, right? They retain the saffron. I'm going to be explaining it in this video, right? So just pay attention. So they have pink color, and then they have outer membrane, and their peptidoglycan layer is very thin. But in the case of gram positive have a thick layer, and then they have purple color under the microscope. So for when you view a gram-negative bacteria under the microscope, it is pink or red in color. But when you view on a gram-positive bacteria under the microscope, it have a purple color. So now let's get into the important part of this topic, which is actually the staining technique, right? So let's talk about the staining, the gram stain. The principle of gram stain. Gram staining. This gram stain, it's made of several types of stain. So let's talk about the first one. The first one is called the crystal violet stain. The crystal violet stain. Crystal violet stain. This crystal violet stain is known as the primary stain. So they have a purple color. So now, when this crystal violet stain is added to the slide where you have placed the material, when this crystal violet stain is added to the slide, it stains the whole slide purple. So all of the material on that slide have a purple color. They have a purple color, right? So the crystal violet stain is known as the primary stain. But now, the crystal violet stain goes and stain the gram positive material and give them a purple color. And also it stain the gram negative material also for this first step, and also give it a purple color. So in this step, all of the material will appear purple after adding the crystal violet stain, because it's the first step. So then after this, we have the next step. The next step is adding iodine. We have iodine as the mordant. The iodine is added to the slide. It acts as the mordant. It just go and fix the slide, right? It helps to fix the slide. So now, crystal violet and iodine will go and make a crystal violet here. Crystal violet and iodine complex to stain the material. So firstly, we add crystal violet stain. After crystal violet, we add the iodine stain, which is the mordant. It helps to fix all of the material on the slide. And then now, the crystal violet and this iodine, they will combine and have this crystal violet iodine complex, which will help to fix the material and make the material more visible, right? Then we also add something we call acetone. The next step is acetone. We have acetone, or at times we use alcohol. We use alcohol or acetone, right? So when we add the acetone or the alcohol on the slide, it goes and wash off the stain. This is one of the most important steps, adding the alcohol or the acetone, right? Because this step helps to differentiate the two materials. So now after adding the acetone or the alcohol, it wash off the stain. Then the gram-positive bacteria, remember they have a thick peptidoglycan layer, right? So they retain the purple color of this crystal violet that we added before. So they have the purple color. After adding this acetone or alcohol on the slide, the gram-positive bacteria will have a purple color. While the gram-negative, we have the gram-negative here. They will become colorless. So after adding your acetone or after adding the acetone or your alcohol, the gram-positive bacteria will retain the crystal violet color, while the gram-negative bacteria becomes colorless. After this, let's go to the next step. The next step is after adding the acetone or the alcohol, now we have the Cantor stain. We have something we call the Cantor stain, and we mostly use saffron. So we use saffron as the Cantor stain. It's known as the Cantor stain. So now this saffron will go and stain the cell wall of the gram-negative bacteria and give it a pink or a reddish color, right? So you see, after adding the acetone, the gram-positive bacteria retain the crystal violet color and become purple in color, right? While the gram-negative bacteria become colorless. So when we add the Cantor stain, which is known as saffron now go and stain the cell wall of the gram-negative bacteria, and saffron has a pink or a red color. So the gram-negative bacteria now retain that color, and they will be looking pink or red under the microscope. I hope you get the concept. So now, right? So let's talk about some example of gram-negative and gram-positive bacteria. For gram-positive bacteria, we have Streptococcus pyogenes, we have Staphylococcus aureus, we have Clostridium tetani. These are all examples of gram-positive bacteria. For gram-negative bacteria, we have Escherichia coli, also known as E. coli. We have the Salmonella species. And another example could be Neisseria gonorrhea. So this is very important for your exam because instructor usually asks for examples of gram-positive and gram-negative bacteria. So now you might be wondering, why are we studying gram staining, or why do we need to do gram staining in the lab? We do gram staining because it helps us to be able to differentiate between gram-positive and gram-negative bacteria. It also helps us in treatment plan to be able to know which antibiotics to give to the patient. Yeah, so it's very important.